I just put new batteries in. So let's see here. I can't believe I got a phone without a speaker on it. So it's all I could get. The line's busy. Well, okay. So really, I mean, this stuff's so important. These videos are just insane. So let's check out another one about the optogenetics, because it is like mind control in the newest way. Like, I've never seen this type of technology. And crap. Didn't do it. That is not copyright. It'll play after great. Alright, let's see. Yeah, so
we've got the entire World Wide Web in our pockets. Chefs have created veggie burgers that bleed like real meat. And scientists can control minds. Wait, what? Yeah, that's not a... I'm Allie Astrocyte, and on this episode of Neurotransmissions, we're going to talk about optogenetics, controlling the brain using light. When I say mind control,
unbelievable.
It might sound like something out of Star Wars, but in the last decade, advances in technology have let scientists figure out how to directly control the signaling between neurons. See, neurons use electricity to send signals.
Looks like people are joining in,
sides of the cell membrane,
and they're getting cut out or something.
Ions,
and thumbs are going up but going down. It's weird.
To send a signal, called an action potential, ion channels
I wonder how many people are really watching.
To allow ions to flow across the plasma membrane, shifting the balance of the electrical charge. This change in the charge sends the action potential shooting down the axon of the neuron, passing the signal along the length of the cell. Since that action potential is the key to how neurons communicate, neuroscientists have been trying to figure out how to control it. Control action potential, and you control the brain. Kinda. But it wasn't until the early 2000s that they found the right tools for the job. In 2003, some scientists discovered a protein called channel rhodopsin, a light-sensitive ion channel produced by an algae. This channel opens in response to light, allowing the algae to sense the direction of a light source, like the sun. Scientists immediately realized that this new protein could be the answer to their conundrum.
Pay attention, because this is your future, guys.
Two separate groups of neuroscientists were able to use viruses to insert the channel rhodopsin gene into neurons. Since channel rhodopsin can be activated by shining a light on it, when it's expressed in neurons, shining a light on those neurons opens up the channels, generating an action potential. Scientists have been able to create mice that naturally express the protein in their neurons. And researchers can surgically insert tiny LED lights or optic fibers into different brain regions to focus their light beam on specific structures or cell types. When they turn on the light, they essentially turn on the neurons exposed to the light. This lets scientists examine how turning those cells on and off affect a lot of different things, including controlling the behavior of an awake animal. And because light is so easy to turn on and off, optogenetics allows very precise control of neuronal activation, like in the realm of milliseconds. Because optogenetics only requires a single gene to create the protein, scientists can target specific types of neurons to make them express channel rhodopsin, letting them pick apart the roles of different cells in the brain. Even more recently, researchers have started using new light-sensitive proteins to influence neuronal signaling. Some of them open and close at different rates, affecting the speed of the neuron signal. And some of them can actually suppress action potentials, letting scientists effectively turn off particular neurons within a circuit.
That is very scary, guys.
Neuroscientists have
With
been able
light.
to discover some pretty fascinating new stuff.
And they don't just have to have you hooked up.
A circuit in the amygdala, sometimes thought of as being the fear center of the brain, that's involved in how we learn to fear something. And they've also been able to tease apart some of the circuits that are affected in movement disorders like Parkinson's disease. Other researchers, including some of my friends here at UCSD, are using optogenetics to understand things like how the brain processes time and how different brain cells are connected to aggressive behaviors. Some scientists are even trying to use optogenetics to help human patients by developing new kinds of pacemakers that use light to regulate the heartbeat.
Oh, it's not a double-edged sword of technology over the mind.
So many different things. The possibilities are really endless. I guess we better be careful and make sure we keep this technology away from the mad scientists, huh? Thanks for watching this episode of Neurotransmissions. If you liked it, give us a thumbs up. And
Wow!
subscribe to learn more
Right?
about the new kinds of technology.
So, yeah, like this is, I mean, you guys see the one where it's like the actual mouse? Watch this. These people are disgusting, like what they're doing to these animals, man. And I mean, what they could potentially be doing to us. I mean, my God, the possibilities are endless, like she said, for this mind control application. Wire up your brain for the World Economic Forum. You guys see this?
I hope to pique your curiosity with a vision to the future of how we might enhance brain function as we know it and possibly change our sense of self. If you imagine the brain as this city, up till now, it's been as if we've been looking at the city from space. We really haven't had the tools to do anything beyond seeing what whole city blocks are doing. So you can see what happens if there's a blackout and you lose a whole part of the city. What you actually want to know is what the individual components of the city, what the people are doing, and what's the information that's being transported
That's
from
what we
one
want
part
to
of the city
know.
to the other.
Yeah, spying everybody.
Optogenetics is the ability to manipulate individual neuronal circuits, refining our vision, the function of individual circuits, and how they relate to different aspects of our behavior and of our personality. I'm going to give you a few examples of what's been done with this technology so far. The basal ganglia is a part of your brain that is damaged in Parkinson's disease. This part of the brain contains many kinds of neurons, and they're all intermingled. If you activate a certain subset of them with optogenetics, you can make a rat freeze up and have difficulty walking. If you activate a different subset, the rats start moving faster. They take light-sensitive proteins from very humble species, such as algae that you find in a pond, and can put them into another animal, such as a rat or a mouse. This gives you the ability to control the activity of your brain cells just by shining light on them. So imagine if we could use optogenetics or a similar technology to get the input from an artificial sensor into our brain. In principle, we could not only restore function, we could enhance our current functions.
Yo,
so we could give
all right.
ourselves ultraviolet light detectors,
Let me
for
put you
example,
on in a
the
second
way
here.
ancestors have. Or we could take an example from fruit flies and give ourselves carbon dioxide detectors.
All right, try to talk
and then
here.
We could automatically sense our air quality. When things go wrong with the brain,
it's a little
just
better.
so
Hold on.
devastating. I think one of the hopes for optogenetics is that if it can work in humans, that it might be used as a tool for restoring brain function.
Not erasing brain function or hindering brain function, right? That's what they're trying to infer, you know, that, well, it is a double-edged sword. So they're admitting that this, you know, optogenetics is, and she's from the World Economic Forum, man. Like, these people have very nefarious plans for humanity. And then so, like the last video, she said, look out for mad scientists. She literally said that. The person studying this stuff, you know. So, like, yeah, it's the potential for genetic modification of large populations, we know, is possible, right? We know that this algae, even, I mean, it's everywhere. This algae that they're saying, if it gets in there, then this type of stuff is possible. Just that. And, you know, the gene therapy, if you have the gene therapy, you're that much more prone to this type of thing happening. And then, so that's the thing with this gene therapy. They're using viruses to actually do this optogenetics. I mean, and then they say, well, we need to target certain parts of the brain for cocaine addicts and stuff like this. And, you know, so what do you think about that? I'm going to try to get this close and just try to talk as loud as you possibly can, because I really have a bad phone connection. You're on live here.
Okay, so that octogenetics shit is crazy, man. I
Yeah.
had no idea they could just use the light like that to like mess with our brains.
He's saying he can't believe that they're able to use the light to mess with our brains like this. And I know. I was watching those weird cartoons with the sirens and all this stuff, and which are like little horror video games. It's where it all comes from, those siren heads.
Yeah.
And which is weird enough that they're putting those into children like animation videos, like the way they're doing with the dancing and all this happy stuff. And then they got the Joker in there on my video yesterday, which is
Yeah, the Joker doesn't seem very like kid appropriate for this kind of use.
right. There's the Jokers right there, Bush and Obama. I got
Yeah.
on camera right now. But yeah, like that kind of, it's a type of hypnosis. It's an archetypal hypnosis. So like they put these archetypes like superheroes in those cartoons because I think it activates more neurons, which makes you more susceptible to the light. I think that's my theory,
Yeah.
right? Is that I was like, okay, is this NLP? Is this neuro linguistic programming? And it could be. So it could be several different forms. There's a lot of different ways to use light in mind control and
So,
sounds and
can I
images.
ask you a question real quick?
Yeah, ask a question. Go ahead.
So, do you think that the idea behind like the programming like the brain is to like make humanity more easy to control for like the world governments?
Yeah, like the whole idea of like the World Government Summit that they just had. The
Yeah.
idea of global government has been around for forever. Just the idea of conquering everything and getting it so you can have people tricked into it is this newer idea, I guess. But it used to be just brute force would be the way to do it, and I think they realized that it'd be a really hard thing to do to be able to get that done in that way. So like Alex Jones or other people talk about like pink tanks, like if Hitler would have had like pink tanks and shot teddy bear missiles or something like that, maybe,
Yeah.
and they shot rainbows out of their butt. Who knows? Maybe that would make it more palpable for the population, so they would accept the globalist agenda, which is really, I mean, ultimately total control. Like they're saying right here, this has been around since like 2003 when you look back at optogenetics. So man, like all this stuff with the World Economic Forum, the Bilderberg Group, all these people meeting in secret, all these smoke-filled rooms where they have these bioethicist boards and they have these different think tanks like the CFR. They're all in there planning out the next year, and so we've seen, by all these different programs on TV that they want us all to hate each other. They want to use
Yeah, they want to like cause wars, you think?
yeah, like so imagine because this optogenetics can influence behavior, right? And Alan Watt used to talk about cell towers influencing people's minds because of infrared microwaves on the brain. Infrared and microwaves, not the same thing. And but like they're different spectrums and they affect the brain a different way. And it always used to be electrical stimuli was the way to actually control neurons, but now we see that no, you could actually genetically alter people with gene therapies by putting these different gene editing software programs basically that are based off of like algae because these different they're almost like logic gates, so they open up and close based off of
Yeah.
the light shining onto it after they're covered with this algae stuff that is receiving light and opening up. So then it actually releases these it causes the neurons to fire. So you can, and it says that they're going to be able to do it in such a way that it would be micro analysis, micro data projection. So you could like have somebody on like a remote control, like an Xbox controller. You're sitting there and you can control people, and it would be have like those VR headsets. What do you?
So
They're
do you
ta-
think the government has like some kind of controller to like get blue light in our brains and like
this,
control us?
this is the interesting question, and from the videos I've seen, I'm going to show more here. The optic nerve also can be used to fire these neurons using this optogenetic technology. So yes, like you can. That's what I was talking about, with, like with the Oculus Rift. So somebody's hooked in, well, you know, they could be under some kind of super MK Ultra. This is basically MK Ultra on genetic steroids. Like, you know what I mean? This is Buck Rogers mind control, and I've never seen Infowars talk about this. I've never seen, and God bless them all, Jay Dyer, all these people. They just did a thing about MK Ultra today, Project Naomi, all that stuff on Infowars. It's like on his channel, he just did a bunch of really good stuff. Yeah, right. So Gemini Moon just said me neither. So this is the kind of stuff I research. This stuff I don't stop digging, man. Like, this stuff is so insane. What do you think about, like, how you've never heard this before?
You asked me from my perspective on having never heard of this before?
Yeah, because I just researched this last night. I just found this out. So I'll admit too.
Last night?
I just found this out last night, and I almost read like an audiobook on it completely all the way through, because I was like hooked.
So
that's why I didn't shave.
I think it's scary, to be honest. I think it's really scary because it makes me think that maybe, like, I like to play video games, right? So like maybe if I had ever gotten like a virus from something that could have contained like controls for my brain, maybe the video games that I'm playing are meant to like control me.
Yeah, and I think that's true. I think any headset you ought to stay away from. Even I'm starting to think maybe even now, from all the stuff I've been seeing the last couple days, man, that I wouldn't trust any of these new movies that come out, because especially like, because you have shedding, you know. And I'm not going to get any deeper. You know what I mean? Like, so who knows if we're infected too? Yeah, headphones too. Gemini Moon they're saying that, yeah, like, this has been around for like 2 decades about now. So who knows what they've been doing behind the scenes with this technology, as far as like having all of Hollywood in on the same page. So you could go back 20 years into Hollywood film possibly and see like this type of, you know, flat type of strobe lighting of different colors, different color spectrums. That could be this ultimate form of mind control, where you have all these different neural triggers for specific neural pathways to stop and turn on. I mean, they're saying they can turn off entire areas of your brain using light.
Yeah, and that would cause a lot of different. That could be a lot of different things they could do with that.
And so Gemini Moon, I just went to the movies. Hold on, I didn't see that comment. You just went to the movies.
Yeah, man, it sounds like the Gemini Moon guy wants to call in. I kind of want to give everybody a chance to call in and talk about this.
Sure, yeah, just, you know, I hope you guys can hear him. Can Gemini Moon comment? Can you hear the caller okay?
It's been nice talking to you. I'll hang up so then Gemini Moon can call in.
Okay, cool, dude. It's really nice talking to you. You're my first caller. This is awesome. Thank you for calling. You're my first caller. Yeah. Sweet, it's legend. You're legend now, dude. This is because no one's freaking looking at OPTOGENETICS on Infowars.
Yeah.
We have to call into Infowars. I'm calling Bart Fine. I'm calling everybody, guys. We gotta call in Alex Jones and tell him to get everybody to do a video about this in the whole Infowars crew. Everybody, we got to get everybody on the same page here, because this guy's talking about gene therapy on here. Like, that's the key to get this going.
Alright, man. Well, I gotta go.
Yeah.
I hope the rest of your stream goes well. I hope you get more of this info out here. You really educated me today.
Dude, thanks for calling. Yeah, that's my goal.
My roommates about this. Thank you so much.
Mad props, dude. You guys are the best. Like, this is what it's all about, man. And I'll go ahead and hang up. That was a great first caller. So you guys can go ahead and keep calling. We got it. So at least it's audible. You know, that's what I really care about.
There's a lot of ethical issues.
There's a World Economic Forum lady.
Your function
Continue
and making yourself better.
Video.
And this is what actually the whole discussion after my ideas lab morphed into, which was not about optogenetics or not what we might do, but whether we should do it.
That's a really great point. So would there be a side effect to increasing your capacity by 20%. It's really, really fun to fantasize about the future. You know, in 10 years' time, we might all be enhanced with
Did you hear what she just said? Enhanced. So maybe it would be like the globalists would be the enhanced people. They would use it for their good, right? It's like that the movie or the video game. I got it right here. BioShock. Have you guys ever played this? Let me get that in there. You guys got to play BioShock to understand the psychology of these people, because that's what it would lead to eventually here. But we're talking about, like, she just said 20%. She was talking about brain capacity, about like increasing your brain capacity by 20%. So like they would give themselves the good stuff, right? And then they would give all the other people. This is just my theory up to just what she just said there. Everybody else, all the plebes, would all get the harmful gene therapies.
That's the future, and it's a future that should be very carefully considered. And I think what we have right now, which is just the possibility of learning, is exciting enough on its own.
Wow! World Economic Forum! The 666 symbol! Unbelievable. So that's just another one. This guy, okay, we have to watch this one, because this guy really gets in depth. He wrote a book on it.
Anxiety has multiple parts to it.
This is old video.
Is used to sort out how all
no,
these
this
different
one's
features
newer.
of anxiety are brought together.
I'm sorry, this
There's a
is from
master
last
control
year.
region that reaches out connections to different parts of the brain and effectively goes and wraps them all up into a unified behavioral state of anxiety.
Of anxiety.
My name's Carl Diceroth. I'm a psychiatrist and a neuroscientist, and I'm the author of Connections. Optogenetics is a really amazing technology, which is a way of turning on or off individual cells.
That does not look
individual
amazing to
kinds
me.
of cells.
I'm sorry.
During behaviors, so that you
That
can
looks
see what
disgusting.
actually matters. It's the complete inversion of how you usually think about using light. We use light, we think, to
For good.
observe things, to observe each
They're using light for darkness, guys.
to observe the world. Optogenetics uses light to control things, to make things happen. And this is really powerful because it lets us ask which are the cells that actually cause things to happen, that actually matter. It's not only a big science aspect, is to understand how the brain works. And once you have a deeper understanding of what actually matters, any kind of treatment can become more powerful. It could be a medication, it could be a brain stimulation therapy, it could even be a talk therapy, combinations of all these. That's the real goal, is basic science understanding. On one level, emotions are subjective, and so we right away have a challenge scientifically, and we'll never really know what a mouse or a rat is feeling, and actually we'll never know truly what another human being is feeling inside. I can only look at ways that
It's called empathy, dude.
and I can try to look for parallels in my own mind. But what we can do is we can indeed do that, and we can look for commonalities, we can look for shared and universal features. And
They're talking about turning people into robots, guys.
Very precisely understand the cells and the connections that cause the expression of these inner states to change, to be manifested, to wax and wane. And we can trace those to their cells and their connections. And a good example is anxiety. Anxiety has multiple parts to it. It's got physical changes where you breathe faster, your heart goes faster. It's also got behavioral changes.
Myocarditis doesn't exist.
Even if there's not an immediate threat. It also feels bad. Also, that's even a third feature to anxiety. It's got this negativity to it. All those different parts, optogenetics was used to sort out how all those different features of anxiety are brought together. There's a master control region that reaches out connections to different parts of the brain and effectively goes and gets those different features that are governed by different other parts of the brain.
They're going after the master controls.
Unified behavioral state of anxiety. That was gives us an insight into ourselves. That was all done with rodents, with mice, but because they have the same brain structures as we do, just scaled down, that gave us insight into ourselves. Another story was I had a patient with autism who had this very prominent avoidance of eye contact, and many patients with autism have this. The question is why, and I in my patients.
Well, the question is, why did they get the autism to begin with?
Give me a very clear description about this. I asked him, does the eye contact make you afraid or anxious? And he said, no, absolutely not. It was not a fear, but he was able to articulate that it was a sense of being overwhelmed by too much information coming in. And that was an amazing moment for me as a physician scientist. Many patients with autism are not very verbal, and that helped us guide some experiments using optogenetics in the laboratory.
Overstimulating the brain with light.
On particular cells and particular patterns of activity that are involved in autism, and to show that there actually was.
Wow, those are real pictures of neurons. That's neat.
Information flow and too much information being not well handled by the neural circuitry that's relevant. So this is how we can come to understand ourselves better, by looking at what optogenetics teaches us in mammals as a whole.
In mammals, like humans.
In terms of implications, first there's the scientific understanding, and the brain has been by far the most mysterious organ in the body. We can look at the heart. It's obviously
humans are the most studied creature in the world,
and so we can model everything we do about it. We don't have that level of understanding for the brain, and so optogenetics is moving us rapidly toward that.
That's their excuse to experiment on us. We don't know what we're doing. Oops.
Work together to give rise to the amazing features of the whole. So that's one implication. Is
don't let any mad scientists get a hold of this.
Getting to better treatments, and again, as I said, even indirectly, that's very powerful. Just knowing which cells to target with any kind of treatment, including medications. And then finally, there's an implication that is pretty important. I would say even philosophically, which is to really know at a very deep level that actions and choices and motivations of individuals truly are governed by just a few blips of activity and a few precise cells and connections. Not to oversimplify things at all, but the fact is we now know unequivocally from optogenetics that adding just a few blips of activity in a few cells absolutely can and does cause very precise, powerful changes in motivation and drives and expression of emotions and all these things that matter so much to us, aggression and parenting and fear. It's philosophically important because it frames these questions of the brain as a biological object that underlies the mind. It makes this very, very clear exactly what this brain-mind correspondence might be.
He's so good at dehumanizing people, man. Did you see what he just did there? Like, oh my God, that's disgusting. Like, no empathy at all.
Evolution and development has been a very long process that's mostly hidden from us.
We don't have any free will.
Our structure and realize that
the human mind is hackable.
The connections we have across the brain, if there's a link between our frontal areas and our brain stem that's governing the expression of emotion, for example, we can ask questions. Why is that connection there? Well, it must be there because it mattered that it was there. Things don't happen and persist for no reason. They can happen for no reason, but they don't persist and grow and survive in evolution for no reason. There must have been some value for that. And so we gain insight into
this guy's so fake, dude.
By looking how we're structured now. It's what a psychopath. Powerful connection with our past. And these connections across our brain, they govern our inner states, and these are shared inner states across all of humanity today. So it's a way of gaining deep insight into who we are and bringing it to this shared physical level. I think it's a very important insight for us to see how these connections inside us also connect us all across space and time.
Blah, blah. What a fake ass, man. What a fake person. So, I mean, isn't this insane, guys? I mean, the fact that they can actually control the brain. I mean, just using light. after they infect you
My
with
name is Stephen Bopa.
gene therapy?
I'm a professor at the University of Illinois at Urbana-Champaign in the departments of electrical and computer engineering and bioengineering. In general, my lab is the Biophotonics Imaging Lab, and we develop new optical imaging technologies and then translate those into clinical applications. So the EAGER project was in response to the U.S. Brain Initiative, and one of the objectives
Obama
that
did that.
President Obama
Yes, we
wanted
can.
through this initiative is really to incorporate new
You didn't do that.
technologies to be able
We
to
made you
better
do that
understand
by
the
controlling your
structure and
brain.
function of the human brain. So we do this with optical imaging, and optics allows us to get that high cellular level resolution. Now what we do is we will feed the light down an optical fiber. So instead of just broadly illuminating the brain with light, we can actually capture an image and be able to see where these cells are and which ones are active and communicating with one another. A lot of this is driven by this field of optogenetics, where it's now possible to change cells so they become responsive to light. And with light, we can turn the cells on and off so they can function in different ways. And that helps us better understand how the neural circuits work.
Yeah, it helps you
And so
better
as optical
understand it
engineers,
by
we have
controlling
this
it.
incredible
You already know how to control
over
it.
the light properties. And we think that by able to control the light properties, we can also be able to modulate or affect the way that cells interact.
They're just tinkering
So
with your
one of our
brain. It's
collaborators
okay.
is Professor Justin Rose.
It's not
who's
like
a
House of a
psychologist.
Thousand
He
Corpses.
also studies behavior and how the structures of the brain relate to behavior in different animals.
Because this isn't electricity. They're using light to stimulate your neurons. So it's okay.
To really connect the optical imaging of cells with how those cells affect behavior. This type of challenge that makes it most difficult for engineers, that the behaviors we see, the outputs from the cells that we're imaging behave in very nonlinear, somewhat unpredictable ways. But that I think is a challenge and an opportunity, because we can apply different technologies to really elucidate or understand how these biological systems work. So that's one of the main technical aspects of this project is can we, instead of changing the molecular biology of cells to make
Yes,
them
Gemini
optically
Moon.
responsive,
That's all they're
and
doing.
we
They're
actually
just
change
hacking
the light
life.
properties to alter the cell behavior in different ways. And it's sometimes
And they talk about
easier to change the
CRISPR
properties of
editing
the light
animals
than it
so
is
that
to
they're
go back and
born
change
with
the
these
biology
in their
of the
brain.
Being
So
able to
eventually they can
genetically
genetically modify
cells,
human slaves that
to
respond to
change
light stimuli. They'd probably just be
function
walking
in living
around with
systems.
those things over their eyes.
Those are great research
And,
tools, but
like,
they certainly are not something we're
You
going to
know,
be
I mean,
doing
can you
in
see
patients
how far
anytime
this
soon,
could go?
if ever. But I think what's interesting is that we can learn a lot from the research that we can do and understand how the brain and the nervous system works. We don't understand how the mind
Thumbs
and
it
the
up.
brain function relate to one another. The brain, when we talk about the brain, it's largely the structure, the neurons and how they're all connected. But there's also the mind and the way that we think and the consciousness that we have. But how are those two related? We just don't know.
Oh,
And that also has
we just don't know.
for health and disease.
We're
brain
gonna.
disease,
There's no
and
spirit.
being
There's
able
no
to
soul.
understand those connections and how those two relate.
We're just
is
atheist
very important
scientist
for society,
gurus.
for our health, and just for science in general.
Yeah, we're just gonna follow orders and experiment with the human beings and tap into their brains because it's good. It's good for you. Now watch this gross shit right here. This is animal cruelty on the
Channel reduction.
umpth degree.
Okay, so which mouse?
And you'll be in these little cells pretty soon, guys, so don't worry.
With AAV6 channel reduction 2 construct. 13 days after injection.
After injection,
So let's
13
start
days.
with this one. This one received a double dose. For now, this is ipsilateral paw.
He felt pain just from light.
So and now contralateral. No reaction at all. This mouse with double dose.
We want total control over the mouse and the squirrel and moose.
No reaction. Ipsilateral.
Ipsilateral.
Next one. So we start from contralateral. Move a little bit. Okay. Contralateral. Contralateral.
apparent node reaction. Ipsilateral.
Yep, there it goes. Big stimuli.
Wow. So contralateral for this one.
Wow. Just from light, it's feeling pain.
Nothing. Ipsilateral. Can you see?
Yes.
It's like that robot. Like, why did you program me to feel pain? You know, it's like, why did you genetically alter me to feel pain from light?
Ipsi. This was the bad injected mice.
It's very easy to tell.
Yep.
Because they all jump and jitter normally.
It's nice to watch the mice jump and jigger around, isn't it, huh? After they take their injections. Isn't that fun? I mean, my God. So, optogenetics is real. It is the new way to control humanity. You know, we just went over this. Here's the end all. I'm gonna end it with this video. This one's really important, and I hope you guys go study all this stuff on your own. But if you want to call in during this, I'll pause it and take your calls too.
Professor of bioengineering and psychiatry at Stanford and the Howard Hughes Medical Institute.
614-372-5102.
Genetics, some of the humble origins of this method in science, some of its applications, and some of the interesting lessons that it teaches us about the process of biology. Now, I'm also a psychiatrist, and a lot of the inspiration for this technology came from psychiatry. And although this is an important field of medicine, it's got a long way to go. In many ways, we haven't progressed too far in our basic description of the core symptoms and their underpinnings, their causal underpinnings, than we had hundreds of years ago. Here's a depiction of some of the earliest medical model type descriptions of depression and this symptom that today we call anhedonia, the inability to enjoy normally rewarding things. And
I think Hillary has that.
The early 1800s described in melancholy what we would call depression today. About as really amazing part of the real forms of life have given us tools that in the last few years have allowed us to come to a deeper understanding of this fundamental and important medical and scientific question, and many others like it. Now,
here we go.
This also gives an opportunity to take a broader view about what is optogenetics, where does it stand in the broader context of interventions, of experiments that we can do in neuroscience. And in the brain, which, as we all know, is a very complicated system with many interconnecting parts, we have historically had the ability to use either magnetic or electrical interventions to stimulate neurons. But one problem is that they cannot discriminate individual kinds of neurons that may be right next to each other. They all look the same to an electrical or a magnetic intervention. What optogenetics allows us to do is to put particular sensitizers, antennas of a sort, for external sources of energy and information into specific kinds of neurons. And then we can use light, in this case, if we make light-sensitive antennas or transducers, and make neurons of a particular kind respond to light. And we do that to allow different kinds of ions to flow into cells and turn neurons on or off depending on the experiment. That's the fundamental idea of optogenetics.
On or
and it's
off,
something people
dude.
wanted to do for a very long time. The first person who seemed to have framed the possible solution to this age-old question was Francis Crick. In 1999, he described the essence of this problem that we needed to control, turn on or off individual kinds of cells, and he suggested the ideal signal would be light. He didn't have an idea of how to do it, but in his usual perceptive fashion, he had some pretty good instincts, I would say. And what I'd like to do here is to sort of pay homage to the many people who have thought about this and done experiments and contributed.
They don't like what I'm saying right now. They just turn me off. Boom.
Making all of this ultimately work. Now, There were early efforts dating back to 2002. A pioneer in this field.
It reminds me of the
Rosenbach, was able to
neuralizer
take,
for Men in Black,
individual
right?
components from a metazoan eye, the multicellular organisms. We all know, of course, we have light-sensitive cells in our retinas, and they achieve light sensitivity with a cascade. He was able to get to the point where he could bring some of those.
It's called a
individual components
phototransduction
of that
cascade.
together and confer light sensitivity.
It's
on
connected to
neurons.
the
And
pineal
bright
gland
areas
too,
of
the
this
third eye.
plot are where the light was on. And there were other groups who also took what you might call a multi-component approach to try to make neurons light sensitive. Now, the only thing that held back these elegant methods was, in many ways, their multi-componency, which contributed to difficulties in targeting. And they, of course, would be the first to state, and in fact showed beautifully that there was some delay in the onset of the responsivity, the action potentials that were fired by the cells in response to the light. And what allowed this
We can't have
field to
that.
take off was the microbial approach, as I alluded to earlier. Now, these microbial organisms, they're small. They don't have a lot of space to work with, nor do they have to do as complex processing as other organisms do. And so they had the leeway, evolutionarily, it seems, to make very efficient, all-in-one, single-component systems for both detecting light and generating ion flow. And that allowed the generalizable aspect of this solution to happen. And this is a field that has its roots even as far back as the early 1970s. Dieter Osterheld and Walter Stachenius in 1971 identified bacteria rhodopsin, and over decades, they and a thriving community of researchers were able to sort out how this amazing protein works, how it responds to light, how its photocycle of the individual protein allows ions to be transported across the membrane. And this pump-type class of microbial opsins, bacteriorhodopsin being an example, was followed by discovery of halorhodopsins, where instead of protons being moved, chloride ions are moved instead. Peter Hagemann and his colleagues, beginning with some early predictive work in 1985, leading to a very important paper in 2002, showed that there were also channel-type microbial opsins that opened actual pores in the membrane instead of pumping one ion at a time. And then many groups here, Feng Zhang in my laboratory was able to find a red light activated channel rhodopsin in 2008, and a great number of different groups. Feng Zhang, Peter Hagemann, our colleague, Ed Boyden, Gerhard Nagel, Ernst Bamberg, John Spudich, and many others have been identifying amazingly diverse microbial opsins with different properties from across the phylogenetic spectrum. Now, what is striking, as you'll notice from this, is how long we've known about these, and
they've been at it for a
this
while.
happens to be my biochemistry textbook, the beautiful book written by Lubert Stryer in 1988. This was the version I used, and you can see this is textbook type knowledge. Description of the photocycle of bacteriorhodopsin, for example, part of the foundational knowledge of biochemists and neurobiologists. Now, there were people who did try to put these microbial opsins in different settings, in fact, successfully. This is a very interesting paper from 1994, where the bacteriorhodopsin was indeed transferred along with light-sensitive flow of ions into a eukaryotic organism. But nobody had yet tried to actually create a tool for neurobiology to turn on or turn off individual kinds of cells. Neurons are complicated, they're vulnerable, they're
that's not true. They had electrical stimuli.
In principle, it was an experiment that could be done, and there were indeed many people trying to do it. Now, the path to this in my own lab started very near to the time when I had started setting up my independent laboratory, and I had
for mind control,
a very humble approach where, as you can see, I used very simple tallies to assess how the experiments were working. And it's interesting to look back on the simplicity of this early experiment. But what I was doing here, I had a broader view in the lab to try different kinds of proteins to see if we could turn neurons on or off with different kinds of stimuli. I had acquired the channel rhodopsin gene. I'd written to a group in Germany that had worked on the initial clone from Garagnago, and he sent me the clone right away, which was very generous of him. I was also looking at different kinds of potassium channels to see
They're
if we
actually
could turn neurons
genetically
on or
altering
off,
these things
constitutively active
so
or
that they
dominant
can
negative
do
potassium
this.
channels.
Optogenetics.
And I was putting all of these into separate groups of neurons in culture to see if I could get control over their activity level. And I was using a very simple, straightforward readout of membrane depolarization, the phosphorylation of a transcription factor called CREB, which I had done a lot of work on previously. And the amazing result was that in these neurons, you can see by the red stain in the nucleus of the cell on a stimulated compared to an unstimulated neuron, both of these were expressing the coupled to a yellow fluorescent protein. So I could see a number of things here. I could see that the gene that I'd put in was being expressed. It was being transported to the membrane, that the neuron was healthy. It hadn't exploded as a result of putting this high levels of this membrane protein into the cell, which can happen, to be sure. And moreover, that the light-activated, the light-treated neuron revealed that it had been stimulated. This membrane had been depolarized. Calcium flux had occurred, and this biochemical readout was telling us that this was happening. And in neurons that were expressing a lot of the channel rhodopsin, I could see a big difference in the number of cells that were reporting on this depolarization event. And overall, it was statistically significant. And although this was a very simple, cheap, humble experiment, this showed us that it was possible. And this was in early July of 2004, but things went very quickly after that. This is what our small group looked like at the time. You can see the two grad students, Feng Zhang and Ed Boyden here, both now at MIT. Feng
I wonder
Zhang
if they're
now
at
also very well
Disneyland.
known for his work on CRISPR-Cas9 systems. But we knew that we had to do a lot more than just show some neurons getting stimulated. We had to show this was unique, that it was potentially generalizable or versatile, that it could potentially be applied.
I wonder how many cartoons have been using
and there
this.
was
Like,
rightly
man,
so a lot of
oh my
skepticism
god!
at the time. But the students.
I don't want to watch anything
did
anymore.
Some really brilliant and creative work. Feng Zhang designed the viral systems that would allow us to put the channel rhodopsin in a very versatile way into different brain regions in vivo. And the fact that it's just a single gene allowed us to be confident that this would work well. Ed did a number of beautiful recordings showing how fast the responses could be, which was another important feature of the system, both its potential generalizability and its speed. And there were many other groups working on this at the time, Stefan Hurlitzer, Zhou Pan, Yao's group in Japan, but we were able to put the different pieces together in time to get the paper out, and it was published together with the two students, Ed and Feng, as well as Gerhard Nagel and Ernst Bamberg. And this, though, was, I wouldn't even say this was yet fully optogenetics, because there was a great deal more work that needed to happen. It took about 5 more years of very intense effort to show that we had actually created something that was generalizable, useful, and broadly applicable, which was the key goal. So optogenetics didn't start overnight.
Applied sciences, guys.
Humble.
That's what it's all
simple,
about.
But it took a number of years to actually say we had finally discovered how to make this work. Part of the problem was, how do you get light into big mammalian brains? And this is an early sketch of Feng Zhang, where he designed the fiber optic interface. And
there's Mickey Mouse right there.
And this looks very much like how we now do it today. So you can see he's a good artist as well as a great scientist. And this fiber optic interface
not a good artist.
Allowed us to
so
do many
basic.
things, but I'll show you one other first experiment. This was 2 years later in 2007. Feng was working made in collaboration with Guoping Feng and George Augustine. And these were mice designed to have channel rhodopsin expressed in the brain. And what Feng did was he implanted into this mouse a fiber optic on the right side of its brain, which, as you know, will control movement toward or attention to the left side of the world. And when he turned on the light, as you'll see here, the animal immediately starts moving and starts turning to the left.
Put on your VR headset.
This was the moment when I would say we finally knew that we had made something that was going to be broadly applicable, generalizable. And that was followed quickly by a collaborative set of experiments with Luis de la Sella, Alex Aravenis, Antoine Adamontidis, and others, where we applied this fiber optic interface to target a deep structure in the brain in the lateral hypothalamus. Fung designed some promoters that would fit into a lentivirus and allow us to control just one kind of cell deep in the hypothalamus. And in this way, we were
Viruses are
able to tie
good.
in certain kinds of activity and show
It's a
some of the
good
neural
virus.
codes that were causal in sleep-wake transitions. And then, even then, though, this required, a very specific fragment of a promoter, the hypocretin promoter, which would fit into a virus. And there was still, rightly so, some skepticism as to how broadly generalizable this would be. But between 2009 and 2010, we finally were able to show that there were a broad array of different tools you could use to generalizably target different kinds of cells. And by this point, I would say by 2010 or so, people were able to see that this could be applied to virtually any system. A good example of how that all worked was in our studies of anxiety. Now, anxiety is a normal part of life. It's a healthy part of life. Of course, it can become excessive.
No, it's not. We have to stop it with all kinds of medications.
But what we were able to do is to use optogenetics to play in patterns of activity or suppress patterns of activity in particular cells and projections in and around structures relating to the basolateral amygdala and the bed nucleus of the stria terminalis. And the way we did some of these experiments is we injected a virus, an adeno-associated viral vector, into the structure of interest, the DNST.
Oh my
But then we
God.
were able to position our fiber optics in other downstream structures. And by doing so, we were able to recruit cells defined by having a particular projection pattern, which, as you can see, is a generalizable strategy. You don't need a special kind of animal or a special promoter. You just need to know your anatomy. And this works because the opsins are trafficked down the axons of the cells, particularly if we help them out with little trafficking motifs that were discovered in the lab by Viviana Gradinaro and her colleagues.
They're helping it.
I'll tell you an example of how this sort of thing works. This is an experiment. I'll play this movie. Kate Tai in the lab did this work, which was published in 2011. This is what's called an elevated plus maze. The so-called closed arm has these elevated walls, and you'll see that the animal prefers to spend its time in the closed arm of the maze, very rarely venturing out into the open arm, which is like walking the plank for an animal. But in our studies of anxiety-related behaviors, what we found and what Kay was able to show—
You need to stay in your houses, guys.
A great deal of other work was that there were anti-anxiety pathways buried within the brain. When the blue letters come on here, you'll see when she started driving one of these, as we discovered, anti-anxiety pathways, and you'll see for the first time the mouse is willing to go out into the open arms and explore them, which it never had done before. Nothing else changed about the animal, the speed, or anything else. It simply was willing to accept this risk, and this was instantaneously reversible. And this, of course, is only one feature of anxiety, apprehension in the absence of immediate threat, but there are other features too: respiratory rate changes, a subjective feeling, anxiety feels bad, and resolution of anxiety feels good. And in later work, we were able to show that different projections coming from the bed nucleus of the stria terminalis recruit different features of anxiety using this projection targeting approach, where we position the fiber optic in a different location than the injection of the virus. And this sort of thing allowed the generalizability of optogenetics to become clear. And so in this sort of work, if you think back to those very early humble experiments in culture, you can see it took a number of years to finally put all the pieces together. I'm often asked, what's the potential clinical impact of optogenetics? Although I'm a psychiatrist, I run a purely basic science laboratory. I'm not working on clinical translation in my lab, but it's a natural question to ask, and other people are working on it. The group of Antonello Bonci found in 2013 that they could turn down cocaine-seeking behavior
Look
in
at
rats with optogenetic stimulation of the medial prefrontal cortex. A very striking—
Look at the human
result
hooked up
in
to it.
2013. And that was then followed up with Anto Bonci and some clinical colleagues with a optogenetically guided clinical intervention. They used transcranial magnetic stimulation, which can be non-invasively delivered to awake human beings. Of course, human brains are a lot bigger, but they have a lot of the same basic reward circuitry. And what they found was, guided by optogenetics, they were able to have a group of cocaine-addicted human beings no longer seek cocaine. They had an experimental group.
I wonder if we could do that with meat, so nobody eats meat anymore. Wouldn't that be great?
Sort of thing shows how
then
far
we could
we've
save the
come,
world
that we can
from global
directly
warming.
affect with a principled, guided approach these very powerful and important fundamental behaviors in human beings. And this makes, of course, coming back to the initial inspiration that we had starting to go down this path, now we do understand in a causal and precise fashion many of the cells and connections across the brain that do affect these very important and both basic and disease-relevant behaviors. Now, this, of course, had very humble beginnings. If you recall those initial tallies in the lab notebook, and we've come a long way since then. But in a way, it's even more striking when you think how these very simple organisms themselves have created these tools that let us make this progress. There's a whole other world
it's progress,
that has
dude.
happened as well.
Total mind control.
How these basic microbial opsins work. We've been able to adjust the speed of the opsins, their sensitivity to light. We've been able to engineer them to conduct different kinds of ions to turn cells on or off, guided by getting to the crystal structure of one of these channel rhodopsins. And we can change the color that they respond to as well. This has allowed us to do something very important, which is to tune the amount and the timing of the activity that we play in to precisely what's naturally occurring in the organism, even that same individual during a very well-defined behavior. And so this,
I wonder if you could do the opposite too.
And precision of the method,
that's what they're saying. Unlimited possibilities.
Think back to how crude and simple some of the early experiments were, and how long it took to get the whole system working. But in the end, that's what makes biology fun and beautiful. And I hope to have conveyed some of that to you today.
There we go. And so I'll finish it off. How about this? With one more, because I had my other live stream, I started out with this one, and I really wanted to get this one on the record here. This one's very much important too. I'm gonna mute that because it's like the stupid copyright crap. But then I'll end it. You guys can still call in. It's 614-372-5102. So let me skip that ad. Let's do this. And I'll end it here, guys. I promise. I just want to get this last one in.
The human brain may not look like much. A couple of pounds of pink, wrinkled tissue. But the last 100 years of neuroscience have shown that the brain is an incredibly densely wired computer made out of hundreds of billions of individual cells called neurons. Neurons communicate with each other using both chemical and electrical signals. When a neuron is activated by chemical messengers, it produces an electrical pulse that results in similar chemical signals being sent to other neurons to which it is connected. Neurons come in thousands, perhaps tens of thousands of different kinds, each with a unique shape, a distinct molecular composition, and a unique pattern of connections within the brain.
It's only 5 minutes long.
For example, these cells, the basket cells, have small input branches and output branches, and they release pulses of chemical that inhibit their targets. Whereas these cells, the pyramidal cells, are large and project long distances, and they excite the cells that they target. If we could figure out the role that each of these cell types
That's those
plays in the
bigger
brain,
ones.
we could understand how they work together to create thoughts,
The smaller
emotions,
ones
and
slow it
behaviors,
down. It's very
and how
interesting if you guys
flaws in
study this
specific
stuff.
cell types may lead to devastating brain disorders like schizophrenia, Parkinson's, and depression. Now, MIT researchers are working to develop tools that allow us to do exactly that. It is now possible to precisely control the activity of specific neurons, and therefore understand how each neuron contributes to brain function. Remarkably, these high-tech tools have come from microorganisms, such as this pond algae called Chlamydomonas. Chlamydomonas is light for photosynthesis, and it swims toward light with the help of a small organelle called an eye spot. The eye spot contains a light-sensitive protein called channelrhodopsin, which responds to blue light by moving positively charged ions across the eye spot membrane, causing the voltage of the eye spot to change, like a battery being charged by a solar cell. But neurons are also electrical devices, and their voltage would also change if channelrhodopsin could be inserted into their membranes and then illuminated. So, researchers took a piece of DNA that encodes for the channelrhodopsin protein. Then, they put it into neurons, using a safe and effective method for delivering a gene, the same kind being used in human gene therapy trials.
Safe and
The
effective.
neurons expressed the DNA, manufacturing copies of the channelrhodopsin protein and installing them in their membranes. As predicted, the researchers were now able to activate these neurons using light, without affecting neighboring neurons. This technique, called optogenetics, allows researchers to drive the activity of specific neuron types to see how they affect brain activity and behavior. For example, researchers can deliver the piece of DNA that encodes for channelrhodopsin into a set of basket cells, shown here in green. Then, pulses of blue light activate only the basket cells, while the other cells, for example the neighboring pyramidal cells, are not affected. So if a specific brain disorder is associated with dysfunctional basket cells, it would be possible to augment the activity of these cells and possibly repair brain function. And these tools don't just activate neurons, they can silence them as well. Archirhodopsin and halorhodopsin, for example, are proteins that are sensitive to orange light and produce the opposite effect to channelrhodopsin. When researchers deliver DNA that encodes for one of these molecules into a specific set of cells and shine orange light within the brain, they can turn off just the selected set of cells. By combining these tools, along with others now being developed, researchers will be able to control brain activity with a degree of precision that would have been unimaginable a few years ago. Optogenetics is currently being used in animal models to understand how normal circuits work, but it may also lead to completely new types of prosthetics for treating a wide range of human brain disorders.
All right, guys, I'll see you soon. I'm going to try to do another stream. It's TBA at this point. Probably maybe even this weekend, I'm thinking. So stay tuned.